Can you launch the introduction presentation? Okay, thank you for joining today. I'm going to do only a very, very brief introduction to the session, and I kindly ask speakers to stick to the 12 minutes, so it's a little bit of time for questions, and then at the end we'll try to have like a general discussion with the audience. And this session is about operationalization of essential biodiversity variables. I believe all of you probably are familiar with the concept, but just in case I would like to stress a couple of important aspects about it. So the EBVs have been suggested as a mean to kind of classify the way how we monitor the many dimensions of biodiversity across space and time. So if you look here to this figure in the left, you can immediately see that there is a huge variability in biodiversity that can be looked from two main perspectives. One is ecosystem level, that is kind of the aggregated properties of all organisms, and the other is species level, yeah? So like, for example, what is spatial variability in the distribution of populations, or the variability in abundances, spatial, genetic composition, etc. All this variability has been, a typology has been derived from this variability by different groups of experts of GEO BON, and this is what we call the different essential biodiversity variables that are classified in different classes. So GEO BON has proposed kind of a high-level definition for each of the essential biodiversity variables, but when you go to the detail, immediately you realize, okay, we need more. We need to understand and define a few more. Oops, I don't know what happened. Can you jump to the second?
The computer. If you pointed the screen,
can you?
like.
All right, thank you. So in Europa One, a project that was built around this concept of essential biodiversity variables to propose a biodiversity monitoring system for Europe, we went much more in detail through those variables together with a large group of experts and define for each of them, okay, what is the specific metric that needs to be monitored? What specific component of that variable, for example, if it is species distributions, what species are important to monitor in order to be to have a relevant information for policy, and so on. So first thing, it's not only about the essential biodiversity variables, it's also about going in deep about how do you define the specific aspects of those essential biodiversity variables that need to be monitored. Second important component is that the essential biodiversity variables very rarely can be developed or monitored by a reduced number of people. You need networks. Networks not only that take the data, but also networks that are able to work together in identifying what are the important aspects to monitor, and also what are the ways of estimating those variables, again, in a way that they are relevant for policies. GEO BON has also established some mechanisms for establishing those networks. And finally, there is a third important component, and this shows the complexity of this component, that is developing workflows for monitoring each of the essential biodiversity variables. This is an effort that we did in a recent proposal that we are doing to the European Commission also for developing an in-situ monitoring system across Europe, taking all the different types of data, of in-situ data, and then grouping them into workflows that then can be used to derive or to monitor essential biodiversity variables. I'm not going to go through all those details, but basically, this session would like to speak and get some learnings about each of those three main components. So we have a number of talks in this session. For example, starting with about BON in a Box, which is a tool that will allow to develop workflows and produce essential biodiversity variables through automated pipelines. To other talks that will speak about those networks that we need to build those EBVs and how to develop those networks. And then some other talks about the specific ways of doing those workflows. So with that, I'm going to introduce first Jordi, and yeah, thank you for coming again, and the floor is yours.
Can everyone hear me okay?
It's for the internet.
Oh, okay. Okay, I'll talk loud. Hi, I'm Jory. Thanks for coming to the session. I'm a biodiversity data scientist working at the GEO BON Secretariat in Montreal. And today I'm going to talk about how BON in a Box can be used to operationalize EBVs through community-driven, reproducible, and scalable EBV workflows. So Nestor pretty much covered my entire intro, so I can just skip that. No, it's okay, I'll give it. Yeah, so EBVs were developed by GEO BON to try and standardize the way that we measure biodiversity across space and time. And there are a set of variables to measure the different aspects of biodiversity from the genetic level all the way up to the ecosystem level. And they're meant to be very flexible, so they're meant to be able to measure across different realms with different sampling types, and yeah, be able to be generalized across all of these different ecosystems. And yeah, so EBVs have gained a lot of traction in the biodiversity community since they were first introduced. Oh, I forgot to mention, so EBVs are of course not raw data. Raw data is very messy. There's data gaps, and there's biases, so it's sort of this modeled intermediate layer that's supposed to describe the state of nature in space and time. And EBVs have gained a lot of traction since they were first introduced, and they've been developed and used in different contexts within the biodiversity community. But there are still a lot of barriers to the widespread use and implementation of EBVs. And the first one is that their flexibility, which is inherent to EBVs, which is necessary, also causes a lot of confusion about what is an EBV and what's not an EBV. The second is because it's not raw data, you have to turn it into a layer somehow. There's not really defined ways to do this. There's a lot of confusion about how to use different sampling types, integrate different types of data across different ecosystems to develop the same sort of EBV classes that still follow this standard. And the last is that, well, not the last, but one of the ones that I'm listing, is that a lot of people are doing this in different contexts, but aren't necessarily talking to each other. So it's not really a standard if everyone's using completely different methods to go from point A to point B. And so now I'll introduce how BON in a Box can help operationalize EBVs and help implement them at a wide scale. So BON in a Box is a platform to integrate biodiversity data and models to generate useful insights into the status and trends of biodiversity. And it calculates essential biodiversity variables and indicators based on a curated open source repository of community-contributed analysis pipelines. And so this is just a schematic of how BON in a Box could fit into a BON, in this case, Columbia BON. So Columbia is collecting data through various means. This raw data can then be put into a BON in a Box pipeline to get EBVs and then hopefully indicators. And a pipeline is just a series of interconnected steps to automate the full process from raw data to EBV or indicator. And then hopefully the information that is derived from the EBVs and the indicators can inform conservation efforts and inform progress and set targets with things like the Convention on Biological Diversity. And it can also help identify data gaps to inform monitoring efforts. So the idea is that it's this sort of circular system that is improving itself and improving the biodiversity information, and then hopefully also informing efforts to conserve biodiversity. And so a little bit about how BON in a Box works. So BON in a Box runs analysis pipelines, which are a series of scripts, and a script is just a single step in a pipeline that does one thing. So it's this modular approach where you break your workflow into these like modular steps that can hopefully be reused in other contexts. So this can be a script written in R, Julia, or Python. It has defined inputs and outputs that are in standard file formats. And because they're in standard file formats, they can be different scripts in different languages can be mixed into one pipeline. I knew that was going to happen at some point. Okay. So each script is described with a YAML description file, which just describes what the script is supposed to do and then also specifies the inputs and outputs and their file formats. And then in Bon in a Box, this generates a user-friendly user form or user interface where the user can specify the parameters that they want to run with the pipeline. So for example, species, country, CRS, any sort of anything that can, any variable that can be defined by the user as defined by the contributor. And then all of these are put together in Bon in a Box by connecting these inputs and outputs that are in these standard file formats to automate the entire process, the entire end-to-end workflow. So from data to indicator or data to EBV. So this is what it looks like in the platform. This is an example of a pipeline. So the user can define different parameters, species, country, et cetera, and then just press run, and it's going to run the entire analysis. As it's running, you can see all of the scripts as they're running. So you can see all of the R logs. You can download the intermediate results. You can go to the GitHub and look at the source code if you want to. So it's all very transparent in that way. And then once you have the final results, they can be viewed on this more user-friendly viewer. So someone who doesn't want to interact with the code at all can just look here and see the final results, whether that's an EBV or an indicator. And then this is more from the contributor side. So this is the pipeline editor canvas. This is where you're writing scripts, you're dragging them into the editor, and then connecting them into full pipelines. And then here you can, there's metadata, so you describe them, you cite them, et cetera. Everything's very well documented. So Bon in a Box can help operationalize EBVs because they allow for these standardized EBV workflows, but with flexibility. So for example, Bon in a Box can take lots of different data sources. Some of them can be global, pulled automatically, such as GBIF, OBIS, Copernicus. But Bon in a Box also can run analyses on your own data, and the data doesn't actually have to leave your computer. So it's really flexible in the inputs that you use. They can be, the pipelines can be generalized to different contexts. But then there's also flexibility within the pipeline itself. So because we have this modular approach, they can be easily customized to specific contexts. So for example, you can add a custom step, or you can remove a step. And then our other sort of pillar of Bon in a Box is that we can use the uncertainty from these EBVs to guide sampling prioritization to then incrementally increase the quality of information that we're getting. And Bon in a Box is completely community contributed. We're hoping to really harness the expertise of the bonds, of the working groups, and be able to share these analysis and decrease the duplication of effort that's happening. We also have these GEO BON working groups that developed the EBVs and developed the classes and developed the EBVs within the classes. So the next logical step would be for those working groups to put pipelines into Bon in a Box or Galaxy or both to be able to share these and for people to use them more. And Bon in a Box is completely transparent. Everything is publicly available on GitHub, all of the code and data, well, unless you're running it on your computer. It's also transparent as the pipeline is running, so you can see the R logs, you can see what's happening. It's not black box at all. And also it's scientifically sound. So we've implemented this peer review process, so all of these workflows, all these pipelines are reviewed by experts to make sure that they're scientifically accurate. So we have a few EBV pipelines available, but we have not nearly enough. So please, if you're developing EBV pipelines, please make them available for people to use on Bon in a Box or similar platforms. And then, yeah, some of our future goals, as I mentioned, we're hoping to mobilize the working groups to get pipelines, EBV pipelines into Bon in a Box. There's also a lot of regional initiatives. Nestor mentioned Europa Bon developed like 83 EBVs that are a lot more specific to their context, and they have all of them on GitHub described. So a logical next step would be to put them in something like Bon in a Box or Galaxy, as we'll hear about later. And then the last thing is that we're hoping to integrate more fully with the EBV data portal so that EBV layers that are calculated with Bon in a Box can then be sent to the portal for people to use. So that's all I have for you. Thank you. Thank you to our sponsors. I'll also say, please come if you want a full demo of Bon in a Box. We have a booth, the GEO BON booth downstairs. So we're happy to do that.
Thank you. We have enough time for questions.
Oh, sorry. I can ask you very directly. So Emma Heslop from IOC, the Intergovernmental Oceanographic Commission. I was wondering how many sort of instances of Bon in the Box there are, and of those, how many are sort of developing marine pipelines?
Yes, excellent question. So as far as instances being actively used, we have a close partnership with the Humboldt Institute, so they have their own instance that they're building pipelines that are relevant to Columbia Bon. Yeah, we have several like collaborators. I'm trying to think of all of them. I'll let Jean-Michel answer this one.
Tobias Sanderman at Uppsala University has one, and we'll test run the HPC integration with him. UNBL, they're using our online instance. So we have an online instance that's linked to a fairly large cloud computer with like 160 gigabytes of RAM, so conduct analysis. They have been using this to run ProtCon for all protected areas connectivity for all countries, which is now in the UNBL dashboard. Marine, do you want to answer that part?
Yeah, I'll also just say that like anyone can go on GitHub and clone it and use it, so we don't necessarily know everyone who's using it unless they're like reaching out to us to collaborate. As far as marine pipelines, we're working with MBON and Ben Halpern on the Ocean Health Index, so we're hoping to get that in Bon in a Box. And yeah, we have no marine pipelines right now, so that's like a big area of priority.
Another quick question here.
Hi, Camila Mazzoni from the European Reference Genome Atlas. What I was wondering is if other initiatives are building pipelines for EBVs, what would be the best way to connect to GEO BON in the Box? Would it be through the working groups? Would it be through some other avenues?
Yeah, so it depends what step of the process you're at. If it's more like conceptualization step, then that would be more like the GEO BON network as a whole. If you like have figured out your EBVs, you've coded them all, you've developed workflows, and you're putting them on GitHub, then that's when you would come to us. And once they're like well documented and separated into steps, it's not so difficult to put them into Bon in a Box. It's more just like, can they be generalized to other areas? Figuring out the limitations and things like that.
Okay, thank you very much. So our next speaker is Ivan Lebras, who will talk about the operationalization of one of those observation networks, in this case in France.
Thank you, Nestor. So it's worse than this morning. I have 45 slides, so I will try to do my best. I want to take a little bit of time to speak about the origin on the French Bon EBV operational pilot, and notably on the Galaxy-Ecology development, because we see that looking at the origin can help us understand, even if we don't know where we go really, we know a little bit of the road we are taking, in fact. So Galaxy-Ecology origin, wow. And I made the things I don't want to make. The origin is a project, a national project in France called 65 million of observers, where the goal was to involve each French guy in science. And I was recruited in the National Museum of Natural History to develop an innovative part to involve citizens in analysis of scientific data. Why? Because of data anatomy, because we need to have power, but in fact, this is not power from IT we need, but power from people, maybe. And to do so, we see that it can be benefiting for the research works. It can also benefit because we can go outside of the researcher's formalism in terms of data analysis. And it can ameliorate global knowledge about environment to better protect it for each citizen who can participate. And the findings we had was we don't want to create something with funny for citizens. We want to first create something where we know that we can really give access to raw data, raw scientific data, and raw source code data, and the analytical procedure from science to citizens. And to capitalize on existing things. So existing things is, for example, Galaxy, who is 20 years old for bioinformatics at the origin. And in 2017, we created the Galaxy for Ecology initiative. We then created the first public instance, European scale instance, Galaxy-Ecology. And then there is a birth of a French biodiversity data hub, PNDB infrastructure, where I was recruited to be the scientific and technical coordinators. And so we decided to take this Galaxy-Ecology things as a brick of the infrastructure. And it was really something benefiting for us because using this kind of platform, existing Galaxy, we directly start in production at production level. We don't have to pay for human resources for sysadministration or things like that. We no need to have IT resources because this is European colleagues who are paying for that. And we just develop bricks when we have money to do that and when we have needs to do that. So it's something quite interesting. The only one duty is we have a minimum quality process in terms of best practices for the tool development and respecting fair principles. So in fact, this is also an advantage. And what was interesting is that the same year when we were in the PNDB infrastructure, the translation of French bon coming to the PNDB, and I was becoming the lead of French bon participation, French participation to GEO BON. And 2 weeks after being recruited, I went to Beijing for the hands-on meeting of GEO BON. Was not knowing so much of GEO BON and things like that. And I remember really that I participated to a BON dev session. I was thinking it was development, like IT development. It was horrible. And just coming from the Galaxy conference from USA, so I discovering BON in a box with notably Maria Cecilia and also Mike Enrique and others who are there, I think. And I just pushed forward this idea also to have something like a sub-part of BON in a box, because BON in a box at the origin is more a toolbox to help creating biodiversity observation network with practice, with documentation, protocols, and a lot of information and things like that. Also a catalog of tools you can use. But the idea was also to say, oh, maybe you can use something that be operational, where you can propose people who take tools and execute it to create workflows or biodiversity metrics. And I think it can be also why now we have BON in a box pipeline. Well, I say that like that, but I don't know. So when Galaxy-Ecology meet EBV to support global indicators, biodiversity indicators production, we need representativeness, sensitivity, who is something we see is inside the concept of EBVs. We need comparability, integration, accessibility, automation. We need reproducibility. We need rigor. And in fact, this same year, 2018, a wonderful paper from Kisling et al. Describing how we can operationalize essential biodiversity variables with its vision of workflow. I was from computational workflow in bioinformatics, so when I discovered that, I said, wow, yes, it is. And there is a mention who was saying, what we need is something like an open source and freely available service in a workflow-oriented infrastructure that supports curation, sharing, and collaboration. This is what is on the text of the article. And I was saying, wow, this is exactly what Galaxy can be and can help maybe in terms of producing accessibility, automation, reproducibility, comparability, and integration. So now Galaxy rapidly is a web platform to share and process research data, permit easy access to cloud analysis and HPC through interface of many programming language. You can push any kind of language in it. There is 4 key principles: accessibility, reproducibility, transparency, and peer review. And briefly, when you have a code, like a Python code or R code, Galaxy allow you to have a web form to execute this script without having any programming language. So from a platform to best practices in biodiversity analysis, so we published this year, after 8 years of work, we take our time, and I'm really proud we take our time, and because when we see the paper, and I invite you to see it, to look at the paper, written by Colin Royot, who was a PhD student with me. It was really a good paper because it is not focusing on technical things. I am a seller of Galaxy, I am a seller of EML, and it's not good for me, it's not easy for me to do so. Colin was really perfect to say, okay, we will write a paper, but around the guidance framework to apply best practice in ecological data analysis, where we push feedback around what we learn with Galaxy-Ecology initiative. And the graphical abstract is there to really summarize the things. We have raw scripts to produce, for example, metrics or indicators from raw data on birds. And we develop what we call the atomization generalization framework, starting to see what are the analytical atoms we can take on the scripts. And after that, look at mapping input and outputs of each of these parts as a workflow, and thinking about generalization at each part and or the entire workflow to apply such a workflow from bird, for example, to other taxonomic extent and or geographical or temporal extent. And at the end, we publish each step as tools in an app store who are reusable to create workflows in existing platform, like, for example, Galaxy. And behind this idea, there is also the idea about taking primary variables from data sets who can be heterogeneous, so we can execute the same kind of workflows to produce the same kind of indicators. Quite briefly, an idea behind that is to have the possibility to have workflow we can easily reuse in 2 or 3 clicks. And to illustrate this, we have this example of the eco-regionalization workflow for Antarctica, where we are taking a workflow originally creating eco-region from project data and in terms of biodiversity and environmental data also coming from the project. And we transpose it in Atlantica on marine fish data sets and environmental data information coming from OBIS. And after that, we can see we can modify modeling parameters quite easily to just test several scenarios using the same kind of workflow. If you want to go further, you can look at the numbers of tutorials we wrote, so you can see what we can do with Galaxy. And quite rapidly, I can go for demonstrating some workflow we have. So something important said yesterday morning by Sabina Leonelli was oriented towards it is essential to track data provenance, data processing, supporting knowledge. And this is concretely what Galaxy can do, tracing the version of the tool, tracing and allowing you to reproduce each tool with the parameters and the data sets who are used as entrants. And it's quite famous to see that I have a picture from the GEO BON hands-on meetings saying something similar around the needs of traceability and provenance of source data and EBV and documentation of production protocol. And so to just finish rapidly, I just see some workflow we have in genetic composition and news from this morning for GNAMO Biodiversity Plus project, where Laura is developing Galaxy workflows to produce, for example, any 500 indicators. So on species distribution and species abundance for species population with marine data, other on breed body survey, on the species traits, on the butterfly, on community composition, where we created a workflow to produce several metrics, and we can apply to several kind of data, from fishing data, real-life survey data, or others. And we can benefit also for a large collection of tools and workflows already existing in Galaxy for this community composition, notably linked to OTU. Ecosystem functioning around the production of indicators on dead wood, for example, and others on indicators for marine data and local indicators. Eco-regionalization in 2 parts, and finally the last one where we were trying to reuse something from Bon in a Box for the red list ecosystem indicator, for example. And I want to just thank you all the colleagues and the young guys who are working with me and supporting me since 8 years to develop this ecology, Galaxy Ecology initiative. Don't hesitate to go on the instance, look at it, test, and comment, criticize, and so on. Thank you.
Yeah, we can see that it's a very active network for that. As you show many EBVs. I'm curious to know if how have you linked, because French Bon is not just calculating EBVs and indicators; it's also collecting data. I'm curious, to know how you linked the data collection with the EBV process.
It's a very good question. I can have several answers. The first, maybe important answer is that, in fact, we are also proposing to share analytical workflow, and in particular EBV workflows, because it's often complicated to share data. So people who have private data can reuse the workflow and produce their own indicators. So in fact, this is flexible, and that's the way we want to go, to not force sharing or transforming the data we want to push into the workflows, but just proposing workflows, and people can do what they want with them. One answer.
You haven't done specific data collection activities?
No, no, we don't want to do so. Thank you.
Then I have one question. So how do you see Thank you. Do you see a competition or a possibility of complementarity between Galaxy and what we have seen before in Bon in a Box?
Yeah. Thank you for the question, because one thing important, it can be something similar. I think we need power to create EBV workflows. As you said just before, we don't have enough EBV workflows, so the important thing is to be interoperable, and so we can populate one or another solutions with the EBV workflows we can have. But the more important thing, separating the both, I think it's complementary, because Bon in a Box was really developed to answer the needs of the EBV things. When we were just trying to reusing existing web platform, which is quite complicated to take into the end, and who is made for something else, to just see if it can fit to the needs of the EBV workflows production. So we see that I think there is complementary. We have already things who can be usable in big infrastructures, and maybe this is something that maybe now, Bon in the Box for now, can be quite limited. So maybe there is something also linked to that we can help. But at least there is exchange to do and to work together in next year, as we are doing, in fact, since months.
Okay, thank you very much. Then from this Galaxy application in France, we are moving to Asia now. Our next speaker is Yayu Takeuchi, if I pronounce your name correctly, probably not. And thank you.
Good afternoon, everyone. I'm Yayu Takeuchi from Japan. Actually, the opposite side of the world, from Colombia, so I took the very long flight it could be the longest flight in my life. But even I was in Bangkok just before this meeting. But on behalf of AP BON, today I will share with you what we have been discussing on the implementation of EBV in our region. So let me introduce AP BON briefly first. So AP BON is a regional network among researchers, government, and organizations across the Asia-Pacific region. And our mission includes to promote interdisciplinary research and to enhance data sharing and deliver knowledge to bridge gaps and inform global platforms such as GEO or EBVs. So actually, last week we had 70th AP BON workshop in Bangkok. In this workshop, we discussed how biodiversity monitoring can contribute to the nature-based solution, as well as way to implement and promote the effective biodiversity monitoring. And in this context, we also discussed how the EBV framework can be applied in this region and what is the challenge to do so. So in the context of the global conservation goals, social demands for the open and standardization biodiversity data are increasing. So however, biodiversity data are often collected and stored locally, and most primary data are described in the diverse or non-standardized formats. In our cases, the data is usually described in our local languages. So therefore, making these local data sets ready to use and interoperable is crucially important. So for example, the concept of EBVs and GBIOS aim to integrate and harmonize the data sets and linking biodiversity information from local to regional and global scales through the national or the regional monitoring networks. So as you know, the Asia, we still face many challenges, such as observational gaps and data gaps and limited data accessibility. So AP BON promotes the local observation networks, especially at the national level, and encourage open data sharing. So in this context, the EBV framework can help integrate local biodiversity data and scale it up to regional to global levels, and to support the design of more efficient connected observing systems. So, and how we can implement the EBV at the national or regional scales? So EBVs are global concept developed by GEO BON, but when it comes to applying them in the regional scale, the several challenges can arise. So at the regional scale, the EUROPOBON here, here provides a very good example of how to develop the regional EBV product across the multiple countries. So so they already developed and defined the 84 EBV data product, and based on the data availability as well as the scientific and social and policy needs. And at the national scale, research groups in several countries such as Finland, New Zealand, Australia, and UK have reviewed their biodiversity data and methodologies to implement EBV written within their national context. So these teams have examined existing data sets and established operational frameworks that effectively apply EBV in the biodiversity monitoring, analysis, and modeling. Then how about the AP region? So I believe there are still many challenges in implementing EBV across this region. So some of which have already been discussed within EP BON. For example, in terms of the data availability, so how much data are available for EBVs? So where are the major data gaps? So in terms of the implementation issue, are the current EBVs sufficient to capture biodiversity trends in Asian context? And how can we collect and integrate the data effectively? And how can we prioritize EBV and develop the national or regional EBV list? And at last, linking EBVs to conservation targets. How can EBV contribute to solve national and regional biodiversity issues? So here I'd like to introduce our recent paper on the national scale EBV to dealing with these issues. So in this study, we evaluated terrestrial biodiversity data sets in Japan and Finland. By the way, one of the co-authors is a veterinary, one of the organizers of this session. I hope he's online. And yeah, so we could compare the terrestrial status of the terrestrial biodiversity data sets for EBV in Japan and Finland. And then especially we compare the Japan data availability with GEO BON and Finland and EUROPOBON, and we identify the unique national data sets in Japan. And then we evaluated how these data sets and EBV relate to global biodiversity indicators and national criteria using example 30 by 30. So as first we reviewed our data sets and portal related to nature conservation and biodiversity monitoring in Japan and Finland. Then we summarize the which of data sets can be used for EBVs. So here is an example of the table. So for example, the dark blue cell like this, it indicates a data set can directly contribute the EBV. And light blue color indicates data set with different primary purposes, but still can be used to calculate the EBVs. So in this way, we identify the connection between each data set and the EBVs, and then one by one. So as a result, so we found 97 data sets from 16 primary national data providers in Japan, and also we found 54 data sets from 14 primary national data providers in Finland, like this. So, and I also briefly mentioned about the data providers, which played a crucial role in securing biodiversity data in national level. So major contributor includes governmental institutions, such in case of Japan, such as BCJ, it's a Biodiversity Center of Japan, or the NIES, it's also the National Institute and the Ministry of Environment. And also national research and data hubs such as Chowta and also the SNet here. So this is a scientific, the group of scientists. So this is also the GBIS conference, and I'd like to mention about SNet Scientific Museum Network. It's a major contributors of JBIF. So in Art's plenary talk on the first day, she mentioned that open biodiversity data from Japan are greatly increased with government-driven efforts. So thank you, Aris, for highlighting Japan. And one of the major projects behind this progress is SNet. So, SNet, so which connects the most public museum collection and try to open the data through JBIF. So including the data from SNet, JBIF has successfully opened more than 30 million records so far, like this. So, and this is coverage of the JBIF has released here. So not only Japan, but also it extends beyond Japan, so contributing biodiversity information at the regional and the global scale as well. So back to EBP Japan. So to identify the unique characteristic of Japanese biodiversity data, we compared data availability among GEO BON and also Eurobon and Finland. And the specific data set for Japan includes phenology, such as cherry blossom, and these are bus drivers and Satoyama index, which represent mosaicness of the land use types influenced by the human activity in the traditional rural landscape in Japan. So these reflect the Japan's distinct geography, culture, environmental context, and interest. So as such, region-specific EBVs emerges when compared to other countries or regions. So I also briefly mentioned we also examined the linkage among the biodiversity among the EBV primary data and also EBV and 30 by 30 indicators in the global targets and national criteria. So as you see, the species population class and also the ecosystem structure widely used in the both global and national 30 by 30 indicators. So this kind of visualization of the data usability is also useful to recognizing which types of data are missing. So here I back to the Asia. So here is the specific issues to advance EBV implementation in our region. So Asia holds world biodiversity hotspots and shaped by diverse climates and complex biological histories. So we so far discussed on these kind of issues, so I have to skip. So here is some example of the SEPA, the socio-ecological production landscape. And for next step is like this, so I cannot read this as I already wasted my time. So, but still, like I shown in the Finland, Japan case, so regional and or country comparisons are a very good way to identify unique characteristics in regions. So I hope we can conduct comparison studies together with regional bonds or country bonds. So I'd be very happy to discuss possible collaboration with you. Thank you.
Thank you for the great talk. Rob Waterhouse from the European Reference Genome Atlas. I noticed when you showed the kind of tables of the two country comparisons as well, and for the first category of genetic composition, there were very few blue squares. And I just wonder if this is like now a priority for the future to try to color those in blue, I guess, in the future for both countries.
Thank you for the question. Actually, yeah, genetic diversity data is most insufficient among the EBV classes. And in case of Japan, we are now studying the project to collect genetic data. So I hope we have more data on this category in the future.
Okay, thank you very much. We'll have time at the end in case you have more questions. But now we are jumping to 4 speed talks. The first of all, the first is from Andrés Suárez Castro on optimizing the use of participatory monitoring data to calculate essential biodiversity variables at local scale.
Hi everyone, thank you very much for coming. So this is a speed talk, so I will be mostly showing just the results of what we've been doing in this project. So I hope you trust me that most of what we've done is kind of robust, but I'm happy to answer all questions that you have about methodology later. So this is a project that comes from a monitoring program that is developed by Conservation International here in Colombia for one corridor in the Amazon. And yeah, and it has like 4 different administrative units, and in this area, it's a relatively big area in the country. And basically what they've been doing is collecting participatory monitoring data for a few years now, and they wanted to know how they can actually use that data to calculate some indicators that will allow them to show that the data that they are collecting is actually doing something in terms of indicating changes in biodiversity in the area. So this is a very challenging process because none of these data is actually structured. They have been just collecting data for different projects. And so, but we know that this is very important because the participatory monitoring data, for example, if you look at the 2 circles there, you will see that the data that comes from monitoring, this participatory monitoring, is actually complementing a lot of what we have in these global databases where we are getting data from. And although they are sending that data to those databases, it's a process that sometimes takes a lot of time, especially when you work with these organizations. So basically, we are working on a workflow that is trying to calculate some essential biodiversity variables. The first thing is defining the scale of analysis. In this corridor, we have a grid of 5 kilometers, which basically was developed after an iterative program where basically we were looking at data availability in the area, and 5 by 5 was the best that we could find in terms of what we had available. And then we basically combine information from this participatory monitoring with some global databases to get information about presence-absence data in the area. So we use a lot of the information from sampling effort, both from the participatory monitoring and the global databases. And we selected only species that have a certain amount of records. So what we are doing here is actually trying to maximize the information from the species that have enough information so we can develop the models. We are not trying to do it for all the species because we know that representativeness is relatively low in terms of the data that we have. So what we are doing here, and it's combining information about the species distribution records with some statistical data, some analysis here to develop these models of occurrence of probability of occurrence for the species in the area. So we combine information from landscape metrics, and then we model the presence or absence for each species in the area for all those species that have enough data, regardless if they are potential indicators or threatened, or if they are in any category that it's important here. So we can develop information about how, when we develop scenarios of restoration, for example, and we link that with the landscape metrics and the models of probability of occurrence, how there will be some increase in the probability of occurrence if some specific management actions are applied at the landscape scale. And, oh, well, how do I go back? And you can combine information from multiple species that have enough data to create these community-level models that will also show you how are the changes. For example, if you have land cover data that is mapped across different times, where you can actually develop scenarios to show how land cover is changing in the area and how that affects the probability of occurrence of the species in each landscape, you can see the landscape where there are more changes in terms of decline or increase of the number of species in those areas. And you can identify landscape with the highest benefits of restoration when you, for example, run different scenarios. If you increase the amount of tree cover or the or reduce edge effects, for example. So this workflow could be used to calculate specific EBVs in terms of populations and communities, and then you can potentially use that information as well for other potential management actions in terms of, and combine it to develop other indicators. There are heaps of indicators that I'm sure you've seen during the conference. So the good thing about this workflow as well is that it's not only focused on finding species in terms of based on the experts and how they think those species are important, but we are also identifying species that are showing responses to the landscape changes that can potentially be common in the area, but are good indicators of potential management actions, and this could maximize the information that we are using. So next steps, so we need to test the sensitivity of this using other type of species as we get more data. I wish I could have changed my slide in the last 10 minutes to say that we don't need to generate these interactive platforms, but that we can potentially integrate this with things like with BON in a box. But the reality is that we actually need these kind of platforms for end users, and I don't know how would be that process in terms of these NGOs and other institutions that are working on their own projects, and where sharing the data sometimes it's more difficult, right? And yeah, we are working already on downscaling into a specific landscape and including other management and restoration actions. Yeah. Thank you.
Thank you, Andres. We'll have, for the speed talks, we'll have the questions at the end. Yeah, so our next speaker is Elnas Neinavas, and it's a record, I believe. And the talk is on linking essential biodiversity variables to GBF indicators through remote sensing for enhanced biodiversity.
Hi everyone, my name is Ella Zainavas and I'm assistant professor at ITC University of Twente, the Netherlands. Today I will be presenting our research on linking essential biodiversity variable to global biodiversity framework indicator to remote sensing for improving biodiversity monitoring and reporting. Here I review two major biodiversity monitoring framework, the Kunming-Montreal Global Biodiversity Framework as well as GEO BON. The GBF provides a structured set of headline component and complementary indicators. These are applied by many countries in order to guide national strategy, action plan, and reporting on biodiversity. Hi everyone, my name is Ella Zainavas and I'm assistant professor at ITC University of Twente, the Netherlands. Today I will be presenting our research on linking essential biodiversity variable to global biodiversity framework indicator to remote sensing for improving biodiversity monitoring and reporting. Here I review two major biodiversity monitoring framework, the Kunming-Montreal Global Biodiversity Framework as well as GEO BON. The GBF provides a structured set of headline component and complementary indicators. These are applied by many countries in order to guide national strategy, action plan, and reporting on biodiversity. Specifically, it contains 4 goals and 23 targets. GEO BON is a global network of researchers that coordinates the collection and delivery of biodiversity information at multiple scales. GEO BON proposed 21 essential biodiversity candidates across 6 different classes. As an important example of development of the EBVs based on Earth observation data is provided by Andrew Skidmore and his colleagues. This study outlined how remote sensing data can be systematically applied to generate EBVs supporting consistent measurement and monitoring of biodiversity as large spatial and temporal resolution. Such approaches are fundamental for linking EBVs to GBF indicator and ensuring a reliable assessment of biodiversity targets. You can see the outcome of Andrew Skidmore and his colleagues on this diagram where they link remote sensing biodiversity product to each essential biodiversity variable under 5 EBV classes. Let's move on to the 23 GBIF targets. Each target accompanied by a set of indicators. In our analysis, we'll focus on how these targets and their indicators are related to EBVs. This diagram highlights how Earth observation and GEO BON EBVs bridge the gap between environmental data and the global biodiversity framework targets. Finally, by linking these EBVs to GBIF targets, we're linking EBVs for GBIF measurable, consistent indicators that can be used for global and national biodiversity monitoring. Moving on to the relevancy of the GBIF target to EBVs, it's important to note that not all GBIF targets and their indicators are relevant to EBV candidates. In this context, a GBIF target is considered relevant to the EBV candidate when at least one of the indicators is conceptually or empirically linked to an EBVs. This definition allows us to systematically identify which targets contribute directly to monitoring biodiversity through EBVs and which do not, ensuring our analysis focuses on the most meaningful connections. Only GBIF target links to EBVs under ecosystem functioning and ecosystem structure have been considered so far in our research. Under ecosystem structure, the identified targets relate to several EBVs, with ecosystem distribution being the most important one. Under ecosystem functioning, the identified targets also relate to several EBVs, with ecosystem disturbances and primary productivity being the most significant one. GBIF indicators rely on EBVs as a foundational input. Increasingly, remote sensing biodiversity products are being applied to generate EBVs efficiently across broad spatial and temporal scale, which in turn allows GBIF indicator to be monitored reliably and systematically. The key references for this study are listed here for your information. Thank you very much for listening. I hope this presentation was informative, and I welcome any comments and questions you may have. I should also add that this research conducted under Horizon Europe Obfuscation project. Thank you.
I'm part of a French Ocean Data Cluster audit. Hello everyone, I'm Clémence Cotten from Ifremer, France. I'm part of a French Ocean Data Cluster auditors, and today I will introduce you the integrated coastal observation system of the research infrastructure EDICO. I would like to thank all my colleagues on the EDICO and Audatis operational team who worked on this project. First, what is EDICO? EDICO is France's multidisciplinary federative research infrastructure dedicated to understanding coastal ecosystems. EDICO is composed of long-term multidisciplinary observation system and community codes, regularly evaluated by scientific peers for accreditation. We have realized that it was difficult to integrate all these data types on a single tool in order to work with all kinds of variables produced by EDICO observation systems. In fact, EDICO includes 9 certified national observation services which observe on the long-term biodiversity, such as phyto and zooplankton, benthic fauna, fish, coral, and so on, coastal dynamics, shoreline, sea level, and physico-chemical conditions in mainland and overseas territories. This project was a collaboration between EDICO and Audatis. The Audatis Ocean Cluster, part of the DataTerra EOSC node, helps the French research community in all aspects of the data lifecycle. Audatis provided its expertise to work with ILICO to standardize data in order to enable interoperability with the aim of being able to combine datasets. The integration challenge was to integrate multi-platform, multi-variable, and multi-scale datasets on a single format and portal. First, the main goal was to improve fairness of every national observation services in order to create data flow from observation services to a single database based on Soundsurfing API standard in order to harmonize all this data. We use common vocabularies such as WORMS and CDataNet and grouping dataset by EBV for thematic exploration. The integrated coastal observation system makes it easier to access and use all ILICO datasets. We developed a user-friendly interface by UX design. The features are access and visualization of time series and spatial data, and cross-analysis of biodiversity and environmental variables. The portal will be officially inaugurated and opened next week during the ILICO General Meeting. Benefits and use cases of this work was to have a useful tool for scientists and decision makers to characterize biodiversity trends and pressure, and to support ecosystem-based management strategies. It's a single entry point to access aggregated EBV data from different sources. For the Audatis team, it was a perfect use case to align complex biodiversity data with interoperability standards such as Soundsurfing, which are not really adapted to this kind of datasets. Thank you for your attention. I am very sorry that I cannot be with you today, but do not hesitate to send me an email for more information.
Okay, yeah. Thank you very much, and thank you actually for being here. And bear in mind, like last minutes, and then we're done. We can relax. So, yeah. So as said, well, let's say this one. Okay. So I'm gonna be presenting this. Just like overall, we all know what the EBVs are, but just to give like a bit of like an overview, like policy-related wise. So one of the outputs, of course, of Europa BON was really to develop the EBO, the proposal for the European Union to really have this like coordination at the European level of, well, what is the Biodiversity Observation Coordination Center. That until now we don't have anything like that. And the call for tenders just finished, so then we are going to have it like for next year, the EBO. So we're going to see how the governance works with all these EBVs, put them into practice in Europe to fulfill the policy needs. As it has been mentioned already, there was 84 EBVs identified in Europa BON. And there is also like 74, for example, biodiversity monitoring programs that it is already seen that reveal some biases already in biomes, taxonomic groups, and its main applications are to birds and habitats directives, one of the two main nature directives that we have in Europe, and the others, the basis of the Natura 2000 as well. So what I wanted to say is like how we evaluated these bottlenecks here. This is a work from Europa BON, and we continued also with BioAgora, the project that I'm currently working on. So there was, we searched for all these existing EU monitoring in situ initiatives that would produce the EBVs. So basically the focus here is more like, okay, we have this list of EBVs, how can we get there, no? What's the information already existing? We're not gonna start from zero, right? So then we created this data set of information. So it was like from the data collection, harmonization and integration level in this database that it's freely available. So you can see all the monitoring initiatives that they are like integrated at the European level in this database. then it was like matching all these in situ monitoring initiatives that they were existing, and then with a focus more of a species population and community composition because of its nature. So for example, giving an example, we have the European Birding Atlas 2 that was published in 2020, and it's had a systematic monitoring of birds like since 2013 to 2017, so very extensive monitoring initiative, like monitor all the birds in Europe basically. And we saw that then, this monitoring initiative can fulfill, let's say, we can produce these marine bird species populations and the terrestrial species populations, EBVs. But then it's like, okay, so how easily can we get there? Is all the information there or not? So then that's the way to evaluate where are the bottlenecks, what are the things that we miss to reach that. So that's, we represent it into like evaluating in 16 categories. So we developed this framework that can be used for different monitoring initiatives and showing it some sort of like a structure of a flower. So every petal is really like one bottleneck that we have to assess. So if there is actually no bottleneck, we have a full petal, and then it's like, full bottleneck is like completely like it's in gray. And then basically, so this is all the initiatives that we could map, and you already can see here what are the biases of all the monitoring initiatives. Basically, you know, we see who are really the winners here and who are a bit like we need actually more in there. So like based on this information, and also you can see how many monitoring initiatives can actually be used for more than one EBV, no? So this was the way to explain like how it identified how these EBVs are. So we are reaching into this like national integration, European integration. What are we missing on the way? No. So this is like a way to identify, to exemplify these monitoring bottlenecks. And what do we have? So positive news. Yeah, we have fewer actually bottlenecks when it comes to data collection. So we're almost there, like in the way the data is being collected. And then some monitoring initiatives are well integrated at the EU level. But then it's like, and some bottlenecks can be tackled together. Okay, so what's the bad news here? Well, so that there is actually limited alignment of the monitoring, like data workflow and the definition of the EBV. And there is insufficient taxonomic, spatial, and temporal coverage. There is a restricted open access to most of the data of those monitoring initiatives, and there is a lacking of long-term funding support for all these initiatives. There is also like a complete automation of the end-to-end data flows. So especially these are really like the forgotten ones, and especially also in marine EBVs. But we have good news, so we have, good news. We have to work on that. It's long-term funding, for example, and coordination hubs. Then we have also like to work as well in the harmonized protocols and improve data integration, but also like as well, not good news, but it's like the collaboration is needed, but we are really working on all these European projects and the coming EBOC. Easy said than done, but we're working on that. Thank you very much. And all
So maybe we can ask to the speakers to come there, and now we have something like 15 minutes to discuss if we want around EBV operationalization. So please, the speakers. Yeah, to make like a panel. Yeah, if you want.
Just gonna move the mic around.
Thank you to start. No? I was thinking you are.
Sorry. Okay. So, very nice presentation, Maria. I really enjoyed it, and many of the others. I'm on the ocean observing side of things, right? And so what I was really enjoying seeing is some of the collection of, okay, where are the observations, and seeing a sort of a data pathway into producing the EBVs. And as a concept, like the ocean observing system, we can't only observe for one thing. We can't just observe for EBVs. There are other applications. So in a way, I think, this is an idea. We see EBVs as kind of one clear service area that we need to deliver to. So how do we get those kind of feedback loops back into the observing system to say what we need?
Yeah, thank you. thank you for the question. I mean, indeed, it's like what we want to get there, and then we can also see now what are the bottlenecks and what is missing. But at the end, we need to come back to actually the initiative, no, the monitoring initiatives. How to tell them like what needs to get let's say better or not. But I mean, in some of these cases, it's like some of the problems might be is like so EBVs are especially explicit also. And sometimes the problems is that the effort, for example, to really get that monitoring effort, it's huge. So in that you need also funding to be able to actually maybe repeat it again in 3-6 years. That's the EBV definition says. So it doesn't also like, yeah, we can also say like, yeah, you have to do it again in 3 years. But then it's like, yeah, with what? Which is the funding? So in a way, this is like, how do we tackle those EBVs like together in a way? Because I mean, in a way, funding can help us also like to reach a better IT infrastructure. It can help us to automatize the data workflows, and then it can also help us to maybe increase again the sampling, no? Again, to do the monitoring. So with this, we are already tackling 3 and more bottlenecks at once. So it's a bit of like, in this sense, it's like providing this framework that can just help us to see what is the big picture, and to really, indeed, like now working with those initiatives. And that's a little bit also like the idea now that we have with this EU project that I'm working on in Biagora. It's like, okay, we have the mapping now, which are the EBVs that can help with policies, but then how do we reach there? And then how we can help like to have this arena of discussion on how do we help getting there by also like tackling all the levels, like from the data, from the monitoring, data collection, so until the very end.
Yeah, I would like to complement a bit or add something additional to what Maria. So one of the very important outputs from Maria's work and colleagues is that there is a lot of data that is actually collected because it is part of all the regulatory process for informing on the state of biodiversity in Europe, but it is stored somewhere in administrations and so on. And very often, like for example, in the Water Framework Directive is like a European level law that requires to collect data on biotic components in the water. And all that data is really, the sampling is really well structured. It's really a very well designed system. But the data is all closed, and it's not, you don't know how the reports are coming from that data, and then it cannot be used for reproducing those EBVs. So even this effort is important just to show, okay, look, you have the data, you're collecting it. If you open it, there is much more that can be done to help you or help others to do those kind of assessments.
Yeah, I just wanted to mention there are a lot of like computational tools that are coming out to prioritize different sampling areas. And I think, as I sort of mentioned in my talk, like the EBV standard can help us understand what we know and what we don't know, and hopefully that can help prioritize different sampling areas. And then when we're looking at EBVs over time, detecting trends, et cetera, we can do like power analyses to figure out, you know, how many samples do we need to detect these trends? Where do we need to go to sample to have a better idea of what's happening? So I think, yeah, hopefully as part of this like whole standardization process, there's also that like prioritization that's built into it.
Thank you. I just had a quick question. I was impressed by the number of sort of data sources that were mobilized in the case of Asia. And I was wondering if you could talk about some of the challenges about how you're able to bring the different stakeholders together to be able to share their data in a common framework. Thank you.
Thank you. Okay. Thank you very much for a very good question. And actually, we are trying to, how to say, engage the biodiversity monitoring community to the GEO BON or AP BON, and it's quite challenging task, but we're still trying to do some capacity building workshop, conduct a capacity building workshop for some outreach activities. And for example, in Japan or the previous EBVs workshop, we are trying to reach out the local students, especially the university students or the postdocs, invited to the EBVs workshop and trying to communicate and trying to engage them how the biodiversity monitoring is quite important for the society as well. Yeah, so but still we are discussing how we can effectively engage these various type of stakeholders.
Thank you. I will have a bit of a provocative question here. You know how I am. Anyway, so the problem is with the EBVs, and then when we go to the CBD indicators that we need to fulfill, let's just get just ecosystem extent. And this is one of the struggles we have. You know, most of the community of EBVs were in the monitoring assessment for EPEs. And then we realized we're chapter 3, so we deal with data and indicators, and we thought, okay, we're going to sort out this with EBVs, but we couldn't because, you know, they're asking us to go into the CBD indicators, which don't match exactly. I mean, we can go through, there's a proxies, blah, blah, blah. But anyway, we need to come up with, show the community a reproducible method. I'm going to take ecosystem extent, which is the simplest one, right? They can be reproducible, they can be, but no, we need to show that. We need to find out a way to do it. And then we thought, well, perhaps we can do a combination of EBVs and blah, blah, blah. And it's difficult. So here I'm happy because I have all the developers, behind, all the work you guys do in Bon in a Box. So are you thinking in how we can move forward, how we can do this matching of EBVs with the requests we have from the community, with the requests we have from CBD, GBIF, etc.
Yeah, I mean, I completely agree. I showed that diagram that's like EBV into indicator, but of course with the CBD, that's not really the case. Like EBVs aren't the base of the indicators. Yeah, most of them not, but I mean, like, I'm not the one who makes these decisions, but I'm hoping that as we standardize sort of like one level below indicator, that it'll be this like iterative process to improve the indicators with the information that we're getting from the EBVs in a standardized way. Because right now it's sort of like very institutionally led, and they have their indicators and they have their data, but if it's EBV based, it'll hopefully like allow people to do it more easily, or allow like governments and countries to have more control over the process. So yeah, obviously, like, hopefully it's an iterative improvement of these indicators as we like, reach new agreements and targets and everything.
A rapid comment, but I will not answer with technical things, but more about the idea about if indicators are relevant or not at the national level, but also at sub-national levels. And I think it's maybe important also to consider looking locally at the territory level, at subnational levels, if the indicators is relevant and if we can produce metrics who are related to the indicators. And if we do so, I think we have metrics who are saying something. And after that, for the reporting, I will say,
yeah. Okay. All right. My question was a bit similar. Oh yeah, I'm Tobias Andermann from Uppsala University. We do a lot of spatial modeling and working with biodiversity data, but I also must say we don't typically think in terms of EBVs up to this point, but I see the value in having standardized measures. So that's also why I'm here, why we are here, to learn a bit more about it. And I'm just wondering, since there's such an overwhelming number of them, and what would you say to researchers, which ones should we focus on, or should we get behind? Are there any of these EBVs that are more related to decision making or to these reporting or indicator frameworks? So can we narrow it down to some that we should really focus on as a community?
So the example that I showed comes from very specific needs, like this NGO needs to basically show the donators that they are actually doing something with the monitoring data that they are getting, right? And we did a review of all the potential EBVs that could be measured in the area, and there were more than 30 or 35 or something. And actually the amount of, the number of variables that we could use was only like 4 of them, right? And it was because of the data that we had, but also because it's thinking about how you can communicate that the data that you're using and all the modeling that you're doing is actually showing a number that they will understand. So I think the concept is very valuable in the sense that it actually helps you to categorize the kind of indicators that you can show. So they are not the indicators, but they can become the indicators. And in our case, for example, we only use 4 of them that were based on the population and the community level. But there are many others that are related with ecosystem integrity or the ecosystem extent, where in many of these areas we actually don't have the data to link like at the resolution that they need, like how the restoration actions are actually, for example, improving biodiversity in the area. So I think it will depend on the region, but I think it's important to move from just the reporting at the global scales and with these big targets and see how these variables are very important to actually track what is happening at local scales and that's the way how they are actually getting the money to continue with these processes anyway.
Because yeah, we can. I can speak a bit of the experience of Europa One. This is an extremely important question, and we did have that challenge, and it took months and years to select a number of EBVs that are policy relevant. So we went through a very thorough process that involved more than 300 experts analyzing policies. And in the European Union, we have one advantage is that there is a kind of consistency in the reporting that the member states have to do to the European Commission. So we could go to the specific policies and then say, okay, what is needed exactly to do each of those reports, and then trying to match the EBVs to all of them. All that process, as I said, took more than 300 experts, and we retrieved more than 80 EBVs. Now, the next thing we did was to analyze how those EBVs are actually mapped with the data that is being collected. And this is a second very big challenge. But my answer would be it requires communication between data collectors, data producers, and experts in policy in order to do this kind of prioritization. And it can be done. I mean, yeah, it can be done. Yeah.
One last question over there.
Thanks. Very nice presentations. I enjoyed every one of those. I like the idea of this cube. You have biodiversity component, you have space, you have time. And this is fantastic if you model and if you want, to scale up and scale out. That works very well, I suppose. My question is how the scales are defined in space and time, and if this cube can cope with the depth in the oceans, with the altitude of the mountains, with the geomorphology, things like that.
I'm speaking a lot, but there's people in the audience also that if you want to take any of those questions. Again, there have been several different initiatives that have attempted to specify the characteristics of those cubes. And again, it's a big challenge. Another example of what we did in Europa One was, again, going through each of the policies and say, okay, what is the spatial scale at which we need the information and how often we need that information so we can set the spatial and temporal dimensions of those cubes. And this allowed us to do specifications for each of those 84 variables, partly accounting based on expert opinion and not so much on analysis, partly accounting also to the potential availability of the data. But again, this, as you were saying very well, this depends also on the applications of the EBVs to the specific problems. I think there are different views of how the EBVs can be monitored. There are kind of global perspectives where you have kind of one for all. Yeah? But I think that there are also many initiatives that are trying to do exactly that. So, okay, so what is my problem? What is the kind of monitoring that I need to do over time, and then select the appropriate resolution and the scales for that. For the second, the one on marine and so on, I'm not an expert, so I'll skip that one.
I can just maybe add that this can also be considered as metadata. There is two things for me. There is this cube ID, but in my opinion, we don't have to restrict it to a cube. It can be, I think we can say also a terasac or something like that, if we have more than, I don't remember. Tesseract, thank you, was presented in Montreal. So in fact, we don't have to limit, because biodiversity is complex. We already said that often. Biodiversity is complex, and we don't need, and we don't have, I think, to limit the manner to constrict biodiversity to something with not fitting the reality of the biodiversity by itself, in fact. So I think if we have more dimension to explore, we need to cope with that. It can be maybe complicated in terms of pure data manner to represent the data. It can be maybe easier if we are thinking about exploring the metadata and having maybe an indirect way to interact with the data from software. That's my answer, but it's just opening the...
No, I think we just reached the time. So I want to thank everyone from being there at this session, and thanks to all our speakers.